So I'm happy to be here today and present to you what we at CEVEC in Köln, Germany are doing. CEVEC is CEVEC is a technology company, and we are dedicated to developing production technologies for efficient production of advanced biotherapeutics. And amongst others, we have for many years been working with protein production and are now producing with partners proteins for cell therapy, for example, high molecular weight proteins like laminins or certain growth factors that are difficult to produce and are required in GMP quality. However, this is not what I will focus on today. Today I will present to you our platform for stable and scalable production of adeno-associated viral vectors. So when, like we've heard this morning in the plenary session, and I'm sure also during some of the other talks, AAV is becoming the predominant vector type for in vivo therapies. However, there are still a lot of unsolved issues around good production platforms for this vector type. So if we look at the current production processes, these very often still depend on inherently growing cells, thus limiting the production scale just by the sheer substrate dependence of the cells. And also all of these processes depend either on transient transfection of 2 or 3 plasmids during each production run, or on infection or transduction with helper viruses. This then in turn is associated with high costs, either for the plasmids in GMP quality or for the helper virus, and in particular there for the analytics and proving the complete removal of the helper viruses. Also, very often the processes are associated with manual handling and thus limited scalability. This is all perfectly fine if you're dealing with an ultra-orphan disease and you have low doses per patient, like for Luxturna, for example, we inject into the patient's eye. But if you think the more recent developments and the projects in development addressing common diseases, addressing diseases where you have to administer the vector systemically, then these production processes really hit their limitations. And this is where our new platform can really help. And this new platform, first of all, relies on suspension cells, so you're not limited by substrate dependency of the cells. You can grow the CAP-GT cells, of which our platform consists, in any standard stir tank bioreactor format, in high-density cultures using serum-free chemically defined media. And much more importantly, we have found a way to circumvent transfection steps or helper viruses. So the system, as I'll explain to you in a minute, depend on fully stable cell lines that can basically be handled like protein-producing cell lines, like you know it from antibody production. And this, of course, then comes with several advantages. On the one hand, you don't require a transient transfection step, meaning you don't have to produce high-quality plasmids. You avoid any potential costs there and also any potential supply issues with plasmid. You don't have to rely on the transfection process, which in itself is very hard to do in a consistent way and which is also limited to a certain scale. You don't need helper viruses. You don't have to remove the helper virus and do the corresponding analytics. And the whole process can be run in a standard stir tank bioreactor process. This makes our stable system suitable for all kinds of different indication areas and diseases, from early phases of the project, as you'll see, to later stage commercial production. So how do we actually achieve this? Everything is based on a fully stable producer cell. These stable producer cells already carry all components that are required for AAV vector production. So they have rapid helper functions, and what's special about them is that we have found a solution to make the rep gene expression efficiently inducible. This has been the trick, and this is also where we have filed IP on, because it's essential to have cells that do express good levels of the rep and helper genes, and you have to do it inducibly because the functions, as you might know if you're working in AAV, are toxic to the cells. Then the final producers, the final producer cells also carry the capsid genes for the serotype-specific capsid, as well as the ITR flank gene of interest. These cells can then be, like any other protein expression cell line, can then be expanded and be in Stirring bioreactors, for example, or other bioreactor formats, and can be subjected to standard AAV production processes. So obviously this final producer cell line is product specific. So we're starting with the cell which is the pre-packaging cell that is the starting point for every single project in which we're working with our partners. And this cell has everything except for the cap genes and the gene of interest. By stably introducing the serotype-specific cap genes, we generate a packaging cell line, and from that packaging cell line, by introducing stably the gene of interest, ITR flank gene of interest, we generate the producer cell. We can also skip this step and basically go from the pre-packaging cell to the producer cell in one single step by co-transfection. We've also been doing this in some projects. So to give you just an example and of some of the projects that we're currently doing, they are with partners, and so I'm not at liberty to share that data, but we have also generated cells just with a GFP gene of interest, just to show how well the system works. So when we screened about 200 clones, we came up with a handful of producer clones that actually yielded in the initial screen between 10E13 to 10E14 viral genomes per liter. And when you look at all the different gene products that need expressed, you see that the helper, the rep genes, and also the capsid are induced to nice levels, functional levels in these cells, and the functionality is then also shown and demonstrated by the formation of full complete AAV functional particles in the cell cultures. So after having shown that the system works, we started to talk to many different potential partners to work with them on their projects. So how is the system being used? Obviously, when you use a stable expression system, there's always a lead time for cell line development, and you might already want some material early on in your project using the same platform, in principle at least. So the way we address this is actually by during the time that the cell line development is taking place here, and that is the cell line development itself, plus stability tests, plus GMP banking of the cells, and so on. We can do initial production runs at smaller scale, even at GMP quality if required, by transiently transfecting in the gene of interest and the cap genes, or if the respective serotype-specific packaging cell line is available, by just the gene of interest to produce material for early phases. So if these early phases are then successful, then you're already set up for really upscaling your production, going into large-scale production for late clinical phases, or even set up for market by using the producer cell line, which in the meantime has been fully produced, characterized, and banked. So the way we work with our partners and clients is we offer services around cell line and process development. So we have partnerships in place with several of the major pharma companies engaged in gene therapy, as well as several of the gene therapy companies. What we're doing is we are custom making the cell lines for them, because we don't exclusively do this. We also allow our partners to do cell line development, but usually they prefer to work with us for cell line development because we have the long time experience at straightforward cell line development using the CAP-GT platform. We will also do transient viral vector production for our partners, either supplying smaller quantities in research quality, or also through cooperating with our GMP partners, which is the Genebet, not too far from here, actually in Lisbon, in Portugal. And also in the US, we have Paragon as preferred CMO partner. They're located in Baltimore at the East Coast. And in addition so we have these partnerships for GMP production. So we'd also do the process development, in particular for the stable cell lines, and also offer some support in the analytical methods. And I should say mainly this is AV projects at the moment. We also have experience with lentiviral vector production, and we have started to generate packaging cells also for lentiviral vector production. AAV is much more demand at the moment, so that's our main activities, but we're also open to work with you if your company should be interested in generating lentiviral vectors transiently, but also using packaging and producer cell lines. And with this, I'm happy to take questions.